Good afternoon, everyone, and welcome to this very special University of Miami Department of Medicine Grand Rounds. Because it's such a special Grand Rounds, we have a special person to make an introductory remark, and that is the president of the University of Miami, President Dr. Julio Frank. President Frank.
Thank you very much, Roy. Good afternoon to everyone. I am truly delighted to join the Department of Medicine here at the Miller School of Medicine to welcome Dr. Anthony Fauci to the University of Miami. At this historic moment, we are proud to be at the forefront of innovation in medical education with our next Gen MD curriculum, which is a path towards the medical profession that emphasizes an individualized learning experience. Its focus on discovery, on the translation of research into clinical solutions, and on leadership developed through mentorship will equip our students to address the many healthcare challenges COVID-19 has accentuated. Our entering class this year is the first to experience this curriculum, but all of our students and residents are members of what we might call the class of the pandemic. Now, we all know that the visibility of health professionals during this global emergency is at an all-time high, and few leaders in our field have been more visible than our speaker today. In fact, we have come to describe the rising interest in the health professions as the Fauci effect. Dr. Anthony Fauci has provided a powerful example as a scientist and physician. He is a towering figure in global health who has safeguarded and relied on the integrity of science without bending to the political pressures and polarization that pose as big a threat today as COVID. As a university president, one of the most rewarding aspects of my role is to champion learning. I am honored to be here today as our residents and students learn from one of the world's best. Welcome, Anthony Fauci.
Well, good afternoon. I'm Mario Stevenson, a professor of medicine and chief of the Division of Infectious Diseases, and I'd like to welcome you to today's Grand Rounds Endowed Lectureship. The Hoffman-Ratson Lectureship was established by grateful trainees of Drs. Thomas Hoffman and Kenneth Ratson, who headed the ID training programs at the University of Miami, Jackson Memorial Hospital, and Mount Sinai Hospitals from the 1970s. Dr. Hoffman graduated from Villanova, received his MD from the University of Pennsylvania, and completed a fellowship in infectious diseases at the University of Rochester. He served as chief of the ID division at the University of Miami from 1973 to 78. Dr. Kenneth Ratson graduated from Amherst College, obtained his medical degree from Harvard Medical School, did his internship and residency at Columbia Presbyterian Hospital. He did his fellowship training at CDC and at New England Medical Center Hospitals, where he was chief resident in infectious diseases. Dr. Ratson came to Miami as chief of the Division of Infectious Diseases at the Miami VA Medical Center before moving on to head infectious diseases at Mount Sinai Hospital. Drs. Hoffman and Ratson were consummate clinician teachers who were revered by their trainees. Their expertise was widely recognized in their community by colleagues and patients. They both set the high standards for their trainees and were considered demanding teachers. This endowed lectureship was established in the hopes of bringing leaders from around the country to share their expertise in infectious diseases, and we are delighted and honored to have Dr. Fauci join us this year. I'll now turn it over to Dr. Gabriela Raffa to introduce our speaker, Dr. Fauci.
Thank you, Dr. Stevenson. It is my pleasure to introduce today's speaker, Dr. Anthony Fauci. Dr. Fauci is the director of the National Institute of Allergy and Infectious Diseases at the National Institutes of Health, where he oversees extensive research focused on infectious and immune-mediated diseases. He has made groundbreaking strides as the longtime chief of the NIAID Laboratory of Immunoregulation, including delineating how immunosuppressive agents modulate the human immune response, understanding the immunopathogenic mechanisms of HIV, and developing life-saving treatments for HIV. Dr. Fauci has advised 7 presidents on HIV/AIDS and many other domestic and global health issues, most recently the COVID-19 pandemic. He was one of the principal architects of the President's Emergency Plan for AIDS Relief, a program that has saved millions of lives throughout the developing world. Dr. Fauci is the recipient of numerous awards, including the highest honor given to a civilian by the President of the United States, the Presidential Medal of Freedom. He is one of the world's most cited scientists and is author, co-author, and editor of more than 1,300 scientific publications. He also serves on the editorial boards of many scientific journals and as an editor of Harrison's Principles of Internal Medicine. Today, he will be giving a presentation titled COVID-19 in 2021, Lessons Learned and Remaining Challenges. Just so everyone's aware, the questions will be by the Q&A tab, so please submit them there, and we will circle through them. So without further ado, I pass the stage over to Dr. Fauci.
Thank you very much, Dr. Rafat, and thank you, Dr. Stevenson and Dr. Frank. It's really a great pleasure to be with you here today to deliver this lecture. As you can see from my first slide, I've entitled this lecture COVID-19 in 2021, Lessons Learned and Remaining Challenges. I have no relevant financial relationships or commercial interests. I'm going to divide this lecture into 7 categories, and I will briefly touch on the high points of each of these. So let's start off with the epidemiology of this most extraordinary pandemic that the world is experiencing. At the end of December of 2019, an unusual pneumonia was noticed in a series of individuals, more than 20, in the Wuhan district in central China. And then literally within a period of a few days to a week, in the very beginning of January, a novel coronavirus was isolated from one of the individuals with this unusual pneumonia and was put on a public database that the world has benefited from with regard to the development of vaccines. Little did we know in that first week in January that more than a year later, we will have experienced in the confines of a single year, the most extraordinary and destructive pandemic that we have experienced in over 102 years, from the iconic 1918 pandemic influenza. As of yesterday, there have been now 107 million cases that have been reported, with more than 2.3 million deaths globally. Unfortunately for us, the United States has been the hardest hit nation in the world, with about 27 million cases thus far and over 460,000 deaths. We have experienced an interesting series of surges of cases over the past year, starting with the late winter and early spring of 2020, when the cases were dominated by the surge in the New York metropolitan area. Note, when the peak occurred, we never went back to a reasonable baseline, and it stood at around 20,000 per day. When we tried to open up the country, some of you may recall opening up the economy or opening up the country again, there were several states that did not abide by the recommendations for gateway phase 1, phase 2, and phase 3. And so what this triggered was another surge in the early summer, again following the peak, never returning to a reasonable baseline as high as 40 to 50,000 per day. And then the most serious of the surges occurred in the late fall and early winter, with a combination of the cold weather getting people to do things indoors, as well as a series of holidays, including Thanksgiving, Christmas, and New Year's. And during that period of time shown on the right-hand part of the slide, we went as high as 300,000 to 400,000 cases per day, and between 3,000 and 4,000 deaths per day. As you can see from the blue line that's diminishing, it appears that we have peaked and are now turning around with the numbers of cases. But with the appearance of variants in our society. We have to keep an eye out on that. And even though, thankfully, we're seeing a diminution in cases, we are not by any means over with this surge. The number of hospitalizations have dramatically increased during this period of time, and in some sections of the country have severely strained the capability of hospitals to keep up. This is a Washington Post article talking about the stress that has been put on certain hospitals and staffs. Examples for, like, in certain of the West Coast hospitals, there were hospitals that had 20 intensive care unit beds, and on any given day, they would have up to 50 patients that would require intensive care. A very severe stress on the symptoms. As you might imagine, this led to a surge of deaths, as shown here as the increasing number of deaths, which at one point, as I mentioned a moment ago, ranged between 3,000 and 4,000 per day. Again, to dramatize this, these are some articles from the LA Times and the Fort Worth Star-Telegram showing that in some locations, they had to bring in refrigerated trucks to serve as temporary morgues since the hospital morgues were overrun with dead individuals. In fact, this is a very striking statistic that the average daily deaths from January 2021, that COVID-19 has been in the United States the leading cause of death, surpassing heart disease and cancer. If you look at the totality of deaths thus far, more people have died from COVID-19 than in the 4.5 years that we experienced World War II, as shown on this slide. There are a number of aspects of this outbreak that are somewhat unique and in many respects disturbing. Paramount among these are the racial and ethnic disparities, where individuals of minority demographic groups, particularly brown and Black people, not only have a much higher incidence of getting infected because of the nature of the jobs that many of them have, putting them out as essential workers in society, exposing themselves to infected individuals, as opposed to the safety of being able to work virtually through a computer. This is a very telling slide comparing the hospitalizations per 100,000 population on a variety of minority groups, Native Americans, Hispanics, and African Americans compared to white non-Hispanics. Note the extraordinary discrepancy in hospitalizations. This spills over when you look at the number of deaths per 100,000. Look at the difference between the 159 for African Americans and the 109 for whites. This has a lot to do with the underlying conditions that are more prevalent in minority populations than in the general population. Let's switch over quickly now to the virology. In January of 2020, a little over a year ago, I wrote this viewpoint in JAMA, and I entitled it Coronavirus Infections More Than Just the Common Cold. I was not trying to be facetious at all. I was just trying to point out to the readers of the viewpoint that we have had extensive experience over decades and decades with coronaviruses. In fact, if one looks at the coronavirus phylogenetic tree, the human coronaviruses are in red font, but the 4 that are yellow highlighted are the common cold coronaviruses that we experience and are responsible for anywhere from 15 to 30% of all the common colds that each and every one of us repetitively experience, usually during the winter months. And then in 2002 and 2012, we had SARS and MERS respectively, the first two pandemic level coronaviruses. And then currently, what we're dealing with now is the third of the pandemic coronaviruses, which we refer to now as SARS-CoV-2. It's a beta coronavirus, an RNA virus, which, as you know, readily mutates, leading to the variances that we are starting to experience with different lineages. There are 4 structural proteins, the most important of which is the spike protein shown on the right-hand side of the slide, which binds by its receptor binding domain to the ACE2 receptor, which is the cellular receptor for coronaviruses on the upper and lower airways, the GI tract, and certainly in other tissues such as cardiovascular tissues. We now have experience with a number of variants which result from the accumulation of mutations leading to new lineages. The one that's most common is the UK variant, which is now beginning to develop prevalence in our own country, such that modelers show that by the end of March, the B117 should in fact be the dominant virus in our country. Fortunately, the vaccines that we've used induce antibodies that still are very effective against this particular isolate. A bit more problematic is the South African isolate, the B351, which is moderately to severely reduced in its ability to being neutralized by antibodies that are induced by the current vaccines, which means that if we have this isolate or this new lineage become dominant in our own country, even though the vaccines would protect against serious disease, such as hospitalizations and death, its overall protection would be compromised. A word or two about transmission. As we know, this is a respiratory-borne virus with predominant transmission by respiratory droplets, usually ones that actually will drop to the ground within 6 to 8 feet. But it has been shown that there is a degree of aerosol transmission by smaller droplets that can remain suspended for a period of time and space. The virus is found in a number of body fluids, but that role in transmission, although unknown, is likely not of any particular significance. Again, getting to things that are very unusual about this virus and makes it unique compared to other respiratory viruses, are the fact that at least 1/3 and maybe up to 40 to 45% of people who become infected remain completely asymptomatic throughout the course of the infection. In addition, when you look at transmissions, 59% of all transmissions come from an asymptomatic person to an uninfected person. 35% from people who are pre-symptomatic and not yet symptomatic, and 24% from individuals who will never develop symptoms. This becomes extremely problematic when you're trying to do identification, isolation, and contact tracing. And in fact, this is unprecedented with any virus that we've ever seen, particularly a virus that has the capability of causing such high degree of morbidity and mortality in certain groups in our population. The fundamentals in preventing acquisition and transmission, we have repeated multiple times publicly over and over. The universal wearing of masks, the maintaining of physical distance, the avoiding crowds in congregate settings, particularly indoors, doing outdoors much more preferentially than doing things indoors, something that's relevant for the state of Florida, particularly Southern Florida, which is much more easy to do this than in states like Minnesota and North and South Dakota. And finally, frequent washing of hands. There are 3 types of diagnostics. a molecular test, namely PCR, that are highly sensitive and specific to give a definitive diagnosis whether someone is infected. Antigen tests are assuming a greater degree of importance, although somewhat less sensitive, when used in surveillance and repetitively can be an important tool in determining the penetrance of infection in any given sector, be it in schools or in factories or in other places where people can gather. And then there are antibody tests which determine whether an individual has been exposed and infected with the virus. There is most recently a home test made by a company, Illum, which is a sensitive, specific point of care test that you can get without a prescription and get the result in 15 minutes. And in fact, the federal government has now contracted with over $200 million to rev up the ability to make these tests to the point where we will be able to produce close to 20 million tests per month. And we believe that this will assume an important role in surveillance. With regard to the clinical course, as I mentioned, anywhere from 1/3 to 40% of the people have no symptoms. Those who do have symptoms, about 80% are mild to moderate, requiring no special intervention, such as hospitalizations. However, about 20% of individuals develop severe or critical symptoms, leading to a wide range of case fatality from 2 to 3% up to 20% in people who require mechanical ventilation. When severe disease occurs, it's predominantly the acute respiratory distress symptoms. However, kidney injury, cardiac dysfunction, and neurological disorders we are seeing more and more of as we get more and more experience with this syndrome. There's a very peculiar hypercoagulable state manifested by microthrombi in small vessels, and even having acute thromboembolic phenomenon leading to stroke, even in individuals with relatively few other symptoms of disease. In some children, there has been described a multi-system inflammatory syndrome rather reminiscent of Kawasaki's disease. We're starting to see some of this syndrome also in adults. Now, who's at increased risk for severe disease? Clearly, older individuals. This is a very dramatic slide showing the difference in the rate of hospitalization per 100,000, comparing younger individuals on the left-hand part of the slide and the elderly on the right-hand part of the slide. In addition, that increased risk for severe illness are people of any age who have underlying medical conditions. And these are some of the conditions that clearly predispose to severity of disease. I point out some that are particularly important, such as morbid obesity, chronic obstructive pulmonary disease, diabetes, and a variety of other conditions shown on this slide. Other conditions which may, not nearly as prevalent as on this slide, but may have an increased risk, are shown here again, just being overweight, again, diabetes, hypertension, and cerebrovascular and cardiovascular disease. There is now being described, and we're learning more and more about it, a post-acute COVID-19 syndrome, or PACS, referred to as PACS, where invariable percentages of people for variable periods of time following the clearance of virus have the lingering of a number of symptoms, including the inability to focus or concentrate, which can be referred to by many as brain fog. In this study from China, you can see that at 6-month follow-up, over 60% of the people had profound fatigue and weakness. Others had sleep difficulties, anxiety, or depression. This is a syndrome which is now being very heavily studied throughout several centers in this country. What about therapeutics? There have been a number of trials. Some have been randomized placebo-controlled trials that have led to the approval of and/or recommendation for use by an emergency use authorization. Remdesivir for moderate to early disease, a number of monoclonal antibodies that have been used both for prevention and treatment. Several of these have received emergency use authorization, as have convalescent plasma. Other interventions are still in clinical trials, such as hyperimmune globulin and a variety of immune modulators. Of note, one of the most effective therapies for advanced disease is dexamethasone in individuals who are hospitalized on ventilators or requiring high flow oxygen. The one thing that we are lacking, that we're putting a lot of effort into developing, are the direct antiviral agents that have been so successful with antiretroviral drugs for HIV and for direct acting antivirals for hepatitis C. In other words, to do targeted drug development in order to give drugs to people very early in infection to prevent them from the need to be hospitalized. In this regard, the NIH has led a treatment guidelines panel, which is made up of individuals with clinical expertise from throughout the country who meet regularly and update the guidelines in real time on the online accessibility as shown here. And new clinical data is put on the website as soon as it is made available, a very important asset for clinicians throughout the country. And then finally, vaccines. If ever there is a success story associated with this disease, it's been in the arena of vaccines, which in fact, Science magazine a few months ago in December of 2020 indicated that the 2020 breakthrough of the year was COVID-19 vaccines. The United States has invested in the development, discovery of, and/or facilitation of the clinical testing of 6 candidates representing 3 separate platforms: nucleic acid in the form of mRNA, adenovirus, including chimp as well as human adeno, and recombinant proteins. There are notable successes already, particularly with the Moderna and the Pfizer BioNTech, which have shown in a randomized placebo-controlled trial of 30,000 and 44,000 people respectively, a 94 to 95% efficacy against symptomatic disease. Of note, the mRNA platform has gone from recognizing a new virus with the availability of a sequence on January 10th, and 65 days later, the product was in clinical trial. On July 27th, the phase 3 trial began, and before the end of last year, in December, the first doses of a 94 to 95% efficacious vaccine began to be injected into the arms of recipients. This is overwhelmingly an unprecedented accomplishment in the field of vaccinology, where something that would have taken years to do was accomplished in a matter of months, less than a year, purely a reflection of scientific advances and the work that was put in for the prior decade for the development of this platform technology. Just to show you how impressive the Kaplan-Meier curve is in both the Pfizer and the Moderna product, comparing placebo with vaccine. These are the New England Journal of Medicine papers publishing, in this case, the Pfizer product with 95% efficacy, and in this case, the Moderna product with 94% efficacy. There are others lined up that will soon be evaluated for efficacy. The Novavax product, which showed almost a 90% efficacy in a UK study. As I alluded to earlier, the effectiveness against symptomatic early disease in the South African variant was only about 50%, but it protected almost completely against severe disease. Similarly, the J&J product, although it was 72% effective in the United States compared to the 94 to 95%, nonetheless, its efficacy against severe disease was very impressive. And in fact, in the study, which included South Africa, there were no hospitalizations or deaths in the vaccinated individuals, even though the efficacy against mild to moderate disease was not nearly as good as the other products. Finally, the issue of vaccine distribution. This is the Advisory Committee on Immunization Practices recommendation for the alignment of how you strategically distribute vaccines. The first group that was phase 1 were healthcare personnel and long-term care facility residents and staff. The 1B was frontline essential workers such as teachers and persons age 75 or older. And some parts of the country are now starting or already have gone into phase 1C, which is persons who have underlying conditions, as well as people from 65 to 70. Hopefully, by the time we get to April, we will reach a point where anyone at any situation in life would have a vaccine available to them. There is a concept of efficacy versus effectiveness. Efficacy is the result of a clinical trial. I've told you that the two that are available are 94 and 95% efficacious. But in order to get an effective vaccine, you have to get it into people's arms. And the reservations and hesitancy, as shown on this slide, about individuals who have concerns about the long-term effects, who have concerns about serious side effects, who have really feeling that they may not get the vaccine, are things we must overcome. Because in order to get the kind of herd immunity, which we'll mention in a moment, we feel you need to get about 70 to 85% of the population vaccinated. We are right now on a route to that, where we are getting close to 2 million vaccines into people per day. In fact, in order to get confidence in the safety and efficacy, as you see from these pictures, the president and vice president themselves got vaccinated publicly, as did myself in the middle, Francis Collins, the NIH director, and Alex Azar, the secretary of HHS. We did this publicly at Mazur Auditorium on the NIH campus. Just a word about herd immunity. We don't know what the percentage that would be required for vaccinating people for herd immunity. That is an estimate based upon our experience with viruses such as measles. But again, the only way to definitively show this is to get to the threshold. And then when you fall below the threshold, you see the breakthrough of infections. Hopefully, if we get 70 to 85% of the population vaccinated, we will reach this level of herd immunity. And then again, there's so many contingencies on this, but hopefully we'll get there by the end of the summer, the beginning and mid-fall. I want to end with this last slide. Having experienced now the third coronavirus pandemic, it is very clear that we might be, and in fact, almost certainly will be challenged again in the future. So one of the most important goals of the research endeavor is to develop a universal coronavirus vaccine, starting off with a universal SARS-CoV-2 vaccine. And as shown in this publication, the time to start is now, and indeed, we have already started. So I will stop there, and I'd be happy to answer questions.
Dr. Fauci, thank you so much for joining us in Miami. These were truly grand rounds in the sense they were both grand and very round, and we are grateful for your participation today. There are multiple questions from lots of people, so thank you for indulging us for a few minutes of your time. Taking up on your very last point about the issue of a universal vaccine for all of the SARS viruses, what is the thinking about the new, these are just variants, or sort of like cousins that you see frequently, but then there are relatives that are developing in various animals like bats and even human primates in different parts of the world that have lots of homologies to the virus but aren't exactly the SARS virus. What's the likelihood that we would ever be able to, or what mechanism should we use to be able to control these other viral outbreaks in the future?
Well, if you're talking about coronaviruses, certainly the bat population and some intermediate hosts are very important reservoirs, and it is very likely that the SARS-CoV-2 that we experienced actually was a virus that evolved from a bat to an intermediate host to the human. So the issue of viruses of different families, what we're trying to do is to develop what we call prototype pathogen approach, where you study an entire family of viruses and develop some commonalities among them, so that when you get hit by another virus within that family, be it a flavivirus, a filovirus, or in this case, a coronavirus, you will have experienced enough with, for example, different vaccine platforms that are easily adaptable to it. So that's one of the things we're doing. And in fact, we're already right now looking at what are the common denominators of coronaviruses are, that we could see if using that as a target for our vaccine, that we might be able to make that first step towards a universal vaccine.
Great, thank you. In another, okay, Mario, go ahead.
Thank you, Dr. Fauci. Nice to see you again, and thanks for that great lecture. A topic that's close to your heart. I mean, we're looking at these variants and worrying about the ability of the vaccines to keep pace with some of these variants. But what about cell-mediated immunity? Do you think that's going to give us a buffer in individuals who get infected, who are vaccinated but get infected? Do you think cell-mediated immunity is going to provide some buffer to prevent disease?
Well, Mario, I think, first of all, that's a great question. I think that's exactly the reason why that when you looked at the J&J trial, which was done simultaneously in the United States and South Africa, even though the protection against infection was approximately 50%, there were no hospitalizations or deaths in those who got infected. To me, that's crying out as a T-cell response. As I think you were suggesting, I think you're absolutely right.
Great. Is there any wisdom at this point in terms of when the precautions that we're taking now, such as mask wearing and social distancing, will be able to be changed? Is it when we reach that 80% immunized number, or is there some other factor that we need to consider?
Well, as I mentioned, that number is purely a guesstimate, but what that number might reflect is when the level of virus is so low in the community that it clearly is no longer a threat, that the RO is way less than 1 and there are very few infections, then I think you could see an appropriate pulling back on the stringency of some of the public health measures, namely mask wearing. Likely restaurants will be able to go back to indoor dining. You likely will be able to go back to a theater, either a theater for a play or for a movie. I think that will happen, but you have to get the virus way below the level it is right now.
When we look at vaccines, is there any scientific data to support the combination of the different vaccine forms or epitopes that they're generated against in combination, or just doing one without the booster?
Well, the J&J, which is right now being evaluated at the FDA and will probably within the next couple of weeks have an evaluation of whether they're going to get an emergency use authorization, is a single dose vaccine. The Pfizer and the Moderna are a prime boost. We know that the prime gives you modest protection that's not particularly durable, but the boost increases the level of neutralizing antibodies tenfold. So in that circumstance, you really want to just stay with the prime boost. If you're asking, is there any way to mix and match vaccines of one type with the other? We don't have any data on that, but some studies are being done to ask and answer that question.
So the neutralizing antibodies that are measured are not the usual antibodies that are done in the general clinical laboratories. How soon do you think it will be readily available to use the neutralizing antibody assay to follow patients that have been vaccinated or previously infected?
Well, some labs like LabCorp already can measure spike antibody. So we now can monitor people and see what the level of their response is to the vaccine. Because if you only have spike but not nuclear protein, that means you got the vaccine, because that's the only protein that is expressed and coded for by the RNA. So I think that in some areas that is already available in routine tests.
What are the lessons, do you think, learned from the recent World Health Organization visit to China? And is there anything that we can take away from that?
Thus far, nothing. I don't mean that in a facetious way. Is that they went there and they couldn't find anything that would really illuminate or enlighten us into what the real question is that what is the origin of the virus? There was nothing that made one suspicious that there was anything other than a natural evolution of the virus, but that's something very difficult to determine.
Based on the data that we have now on the phase 3 studies for both Moderna and the Pfizer vaccines, is there any indication that one would need another booster a year afterwards, or is it too early to tell?
Too early to tell. A good question, everybody wants to know the answer to that, is what is the durability? We know that it's at least 6 months. It may be much, much longer, but clearly it's at least 6 to 8 months. But you don't know what it is until you follow people out for a couple of years.
So here's a question from one of our fourth year medical students who's just about to start her residency in internal medicine. What advice do you have for graduating medical students who will be starting their careers in the middle of this pandemic?
Well, I mean, it is an extraordinary opportunity in the sense of you're entering the field of medicine when a medical issue, historic pandemic, is dominating our world. So, I mean, if ever you wanted to be in a vocation, as it were, that is highly, highly relevant, medicine is the one, particularly if you have any interest in infectious disease.
I knew you were going to say that.
But I mean, if this individual is graduating and going to take internal medicine, that person is going to get an exposure to all of internal medicine, which is obviously very critical and important for your overall training. You have to do internal medicine before you do infectious disease.
That's great. Absolutely. And in terms of the concern that people have if they get vaccinated, that we would be selecting for a variant virus that isn't necessarily susceptible to that vaccine. How real is that concern?
Well, it is a concern, which is one of the reasons why you really like a vaccine that is 94, 95, 96% effective, because the more effective it is in preventing the replication of virus, the more likelihood it's not going to mutate. It's the same thing we experienced with antiretroviral drugs. You know, if you get a combination of drugs that dramatically suppresses virus replication, viruses can't mutate if they don't replicate. So if you suppress the spread of virus in the community with a really, really good vaccine, the likelihood of your getting mutants is going to be very low.
Good. When do you think the data will be available to safely immunize children under the ages of 16? For which,
well, that's a great question. I just actually answered that question in a White House press conference that I was on about an hour ago, but I'll answer it for you all. Right now, we are beginning what's called age de-escalation studies, where you take children from 12 to 9, and then 9 to 6, and then below 6, and you do phase 1 and phase 2A studies for safety and immunogenicity. You don't need to do a 30,000 or 44,000 person trial for efficacy. You just need to bridge the data to show A, it's safe, which it likely will be, and B, that you can induce a comparable immune response that you know from the adult studies are going to protect you against clinical disease. I would imagine that we would likely, by the time we get into the summer, late spring into the summer, that those studies will have been completed and we could go ahead and start vaccinating children as we get into the late summer and early fall.
So the pandemic clearly has had ramifications on every aspect of our society, and from the economy to the way in which people interact to the psyche and so forth. But specifically with regard to our research agenda here in the US, and those of us in the audience that are dependent on NIH funding for doing the things that we did before the pandemic, how do you anticipate the response will be in terms of funding and research to further look at the things that we were looking at, although they take one obviously a different perspective after the pandemic?
Well, you know, it's very clear that the research enterprise has taken a big hit over the past year, and that many investigators have been unable to carry out their research because of lockdowns and the stress on the system. In that regard, what we're trying to get funding for, to the tune of multiple billions of dollars, is to kind of get people back on track again who've lost a considerable amount of time, and really get funding for people to get their programs back up and running, because several of them have been able to switch over to COVID-19 research, but others, where they were not equipped to do that, really had to slow down greatly their research program. So there's a lot of catching up to do right now. Hopefully we'll be able to do that as we get into the fall, and there are very, very few cases that are currently around.
So a stimulus package for scientists.
No, it is. That's exactly what it is.
Yeah. So Dr. Fauci, you're terrific. You need a vacation. And I can't think of any better place than Miami. So when this is all over, we welcome you with open arms to come visit with Mario Stevenson and all the wonderful ID people, and with our president, Frank. And we would welcome you with open arms. You've been very gracious with your time, and we're very honored to have someone of your stature come and address us at Medical Grand Rounds. Thank you, everybody, and thank you, Dr. Fauci.
Thank you very much. It's a pleasure to have been with you. Thanks, everybody. Thanks, Mario. Thanks, Julio. Good to be with you.
Thank you very much, Tony.